Hi, this is Rich Riccieri. Today I'm going to talk to you about the road to commercialization, a CDMO perspective. I'm the Chief Operations Officer of Avid Bioservices. We deal with the contract development and manufacturing of biologics. So a little bit of our track record here at Avid. We've been manufacturing biologics for about 27 years, since the early 90s. We have both early phase and late phase projects, including commercial biologics that are actually approved in 90 countries. We have a lot of projects where we proceed into process validation. So we have 10 projects in process validation, went through PAI inspections, 2 commercial products, and we've been very successful with the FDA. Over the last 4 audits, we've had 0 FDA observations. So a little bit about Avid Bioservices and our scale. We have 2,000-liter single-use reactors. We have 3 units of these sizes. We have 2 1,000 liters and a few smaller reactors. Everything is single-use and disposable. At this time this year, we are in an expansion phase where we will add more units of that size. In this case, 6 2,000-liter subs will be added to our facility. So we will be one of the larger CMOs in terms of capacities and cell culture volume. So we are fully committed to being a disposable manufacturer, and we will always be committed to doing cell culture manufacturing of biologics. So one thing I want to talk today, again, is in the CDMO world, it's very important to try to get a one-stop shop to take you from early stage to late stage. And I'll go through the road to commercialization in a minute. But meaning that the decisions that you make at small scale at the early stage can have very important effects late stage. But in general, having any CMO that can take you from the very beginning to the commercialization is very important. You'll have shorter timelines. Process knowledge will be contained within one organization. And most importantly, you would eliminate site-to-site tech transfer programs, or site-to-site, which would entail site-to-site comparability studies. So having one site, having one CDMO that can do all of that is very beneficial, especially in this case where time to market is critical. So again, the shorter timelines, the time to market is very important to having one CMO that can do everything from development to commercialization. Okay, onto the road to commercialization. We'll go from the very beginning of the process to the final commercial process where you do commercial monitoring. So the first stage, well, before I get to the first stage, the process validation is very important to understand. It's not just doing processes at large scale and saying everything's okay. It's actually a program from the very beginning of process design, which happens in the very beginning of early phase one projects, all the way to the end of phase 3 in commercial production. And through the FDA guidance, process validation is defined as the collection and evaluation of data from the design phase, from that early phase, through commercial production. So it doesn't say up to, it says through commercial production, which you're trying to create a technical basis of evidence that the process is capable of consistently meeting your standards. It takes place, this process validation mindset should take place over the entire life cycle of the product, not at the end of phase 3 where you're looking at process validation and qualification at scale, but from the very, very beginning. And so some of the things we'll talk about is our approach to process validation, but in general, what the industry is doing from the very beginning of the process design. Essentially at this stage, you're really building and capturing knowledge. And you're trying to figure out, at least at the rudimentary stage, what kind of process control strategy you want to employ. And you would then have the basis of a process to then optimize and refine later on. But we want to establish some basic control strategy at this stage. So for process design, essentially, you want to create a strategy where you at least have some process knowledge. We're talking about the phase 1 projects, the early phase projects. You don't have time to do full development. And of course, it's not ready for process validation, but you want to do some very basic studies that are very important to understand. So your cell culture expansion criteria, how well your cells grow, how they cut back, what's your viability profile. Your bioreactor controls, of course, are very important. You want to have as wide controls as you can, wide parameters as you can, but you really want to have an understanding of your temperature, DO, pH, all these set points, all these parameters, harvest viability criteria, really understanding in a very basic way what your process is allowed to do. You want to have a good understanding of how well your cells grow, and if there's a product quality profile during the course of the run. Does the product look the same on day 3 of your reactor as it does on day 10 or it does on day 14 when you harvest? So having that basics is very important at this stage. And going on to the downstream on the purification, you really want to have an understanding at this point of what is your load density, how much product you can load onto a column, when do you collect the product. The very important is your peak criteria, when you start and stop collection. Have a good idea of your impurity, your process-related impurity removal, your DNA, residual protein A, for example, how that gets removed, and your product-related impurities like percent monomer, aggregates, anything that, any smaller species. Really want to have a good understanding of that. In phase 1, of course, you want to really have a good understanding. You have to do a very basic viral inactivation and viral clearance studies, usually 2 model viruses in duplicate, and you want to show that you have a good orthogonal methods of removing and inactivating viruses at this stage, that they're very robust, and that's something that you can keep as this product gets moving forward into phase 2 and phase 3. You want to keep an eye on your flow rates, making sure that your product is going through the column at an acceptable flow rate. The residence times are equivalent between what you've done in PD versus what you're doing in manufacturing. So these are basic ideas, but you want to have that basis as you go forward. And typically to do this type of work, you need a very extensive process development lab, like the one at Avid, what's shown here, where you have reactors that are at small scale and can be done at large scale. But the important point is you can do these process development, you can do DOE studies, you can do at least have some apples to apples studies where you can say all the reactor conditions are the same except for this 1 or 2 parameters. And at Avid, what we do is that we try to make sure that every reactor configuration is the exact same. So we have identical bioreactor controllers and bioreactor units themselves. And when you do this type of work at a small scale and you understand the kinetics and some of the mixing characteristics of different size reactors, you can then scale up your process from the 2 to 10 liters into the pilot scale of 50 to 200 liters, which then gets you on your way to the manufacturing scale of 2,000 liters. So it's very important to do these types of range finding studies, I would say. And you end up with some diagrams like this. Is a DOE space, DOE study, where you can really identify some of your optimal conditions for growth, optimal conditions for productivity. You have a data set that shows you ranges. So in your manufacturing scale, you want to kind of be within these ranges. When you do manufacturing batches, you typically only do 1 or 2 at a time, or maybe a few per year. So you want to compare that to a very healthy data set. So you can say, okay, this is fairly normal for this process to be within this range. So not everyone can do a DOE study, but if it's available for you to do some sort of range study, so when you do the manufacturing for phase 1, you then have an idea of how well you're doing compared to the small scale design space that you have. Okay, so that's really getting you the bones of the process. You get into the clinicals with this type of process, and then if the process or if the project succeeds into further clinical trials, that's where you do a more deeper dive into the process qualification, which leads us to the next set, is when you do work at scale and how that relates to the process validation. So in this stage, you're really doing a deep dive into the process itself. You want to use your final scale process. You want to do risk analysis of your facility, equipment, and design, your utility qualification. You want to have pre-approved protocols with predefined specifications and acceptance criteria. And then, of course, you want to execute at this scale, all the while taking some extensive testing at different time points, looking at every critical item that you can think of during this time. So you're really at this point in the process qualification, you're challenging your process very extensively. You're really trying to find its weak points at this point. And at that point, you want to say that your process is validated, but you really want to have a very, very extensive challenge against the process. So again, it's not just running the process at large scale. There's a lot of support studies that have to happen at this time when you want to validate a process. From upstream point of view, you want to show that your media mixing times are acceptable. You want to show that your end of production cells are stable and they don't cause any problems. So you want to take samples of your cells at the end of a process and keep them going for a few more generations to show that they're stable. You want to show that your limits of inoculum expansion are robust. You don't want to grow them too high or too low when you transfer. Same thing for downstream and support. There's a lot of these support type of projects that have to go into a process validation plan. So how you mix your buffers, extractable and leachables of your product contact film, any sort of carryover, process hold times, anything to do with membrane sanitization, resin lifetime, buffer hold times, virus validation. At this point, you're going to be doing 4 viruses in duplicate and really challenging your downstream process to remove and activate and inactivate viruses. And the same thing for the supporting activities, your final container closure studies, shipping validations, freeze thaw, your bulk, if that's something that's important. And of course, all your raw material evaluations for this. So all this has to be combined into one process validation program. So the next few slides show a few examples. In general, what's very important when you do a performance qualification at scale, when you run your process at scale, is you want to make a list of your critical process parameters. These are called critical because it affects the product quality. And what's very important to understand is you really only want to say something is critical if you are certain about it. You don't want to make a guess. You have to have data or some other method to be absolutely certain that that is a critical process parameter. If it's not, you would call them a non-critical process parameter, which is really more for, could be affecting yield or growth of the cells, but does not pertain to the product quality. So making very sure that you list appropriately these parameters is very important. And then of course, when you execute your large scale, you're keeping an eye on these process parameters. And then you're also looking at the output of the product itself, your critical quality attributes and acceptance criteria. And again, these only have to be certain about that in your program. So at the end of the day, you have to be very careful calling something a critical, because if you have a critical process parameter and your process doesn't meet that parameter for whatever reason, even transiently, you could reject the whole batch, and then you have to justify and then restart the process validation program. So these are very critical terms to use and only to be used when you're certain. Here's another example of a process activity during process validation is the characterization of your resins between fresh and used in its ability to remove viruses. In this case, we're looking at fresh and used resins and 2 model viruses in duplicate. The used resins are 57 cycles old, and you're seeing equivalent performance. So you really want to challenge the resins at both fresh and used for your phase 3 process validation and really show that they're equivalent and comparable. Your resins don't lose its ability to remove the viruses from the process, even after being used for 57 times. So this is something that's typically simulated and done at small scale. So you'll take resin, you would simulate a run, 57 runs, and then you would perform this type of study when you challenge the resin with viruses and then determine how well it removes them. Some other projects that you have for the process qualification. Again, you want to have preset criteria for viability and growth and productivity. You want to set them fairly wide, or at least as wide as you can at this time. You still probably don't have enough data to really narrow it down. Maybe after a few years of commercial, you may have to narrow it down. But for process qualification point of view, you can probably keep these parameters and these specifications fairly wide. And just showing some of the control charts that you have here, I mean, showing 3 or 4 runs that have the similar profile, similar peak viable cell density, similar titers is really enough that you need for process qualification. A few more activities I'll talk about. One is the extractable and leachable studies. Typically, more and more processes are being performed using disposable units, such as reactors or in-process bags. And what you need to do is look at every product contact surface that the product sees in its film, and using some of the USP guidances, you can make a scoring of each one of these films and then determine how, the risk of each of these to the process. And typically for proteins and that type of processing, the risk is typically very low unless you use some very extreme solvents or very, very low and high pHs during the process. Then the scores would get higher, but typically these scores would really remain fairly low for biologics processing. But it is an exercise that you have to do and you have to show. Another project you have to do is to show that there's no carryover in your large-scale purification columns. Typically, that means that after a manufacturing run is performed, you would then simulate a run just using the buffers with no product. And then you want to load, simulate a load, simulate wash volumes, simulate elution volumes, and collect that elution using guidances from the manufacturing run itself. And then you want to check those eluates for process-related impurities like host cell proteins or residual DNA. You really want to show that there's very minimal carryover from one batch to the other. A couple more qualification tasks. Again, this was touched upon before. You want to take some feedstock from your large-scale run and simulate small-scale work in your columns to show that the resin can handle many cycles. You test for impurities and tighter recoveries and yields, and the same thing that you do at manufacturing scale. Finally, the last step, one of the last projects to do is really want to make sure that your in-process holds are as long as possible. Typically, that would mean taking some feedstock from GMP and storing them in small-scale bags to determine how well the product and the film interact. Typically, a 7 or 14-day study showing that the product has no issues and that there's no extractables coming from the bags into your protein is just very important. Typically 3 or 5 days showing that the product is sterile and there's low bioburden is also very important. So this type of program altogether shows that when you hold product in bags, nothing bad will happen to them, and you can have these fairly long, if you need them, hold studies during the process, which may be critical on some days if you have to wait for a piece of equipment to be open, or if you just need to have a stop in the project for a few days. You have the data to support that. Finally, the last lesson learned is from these process validation programs, there is just a tremendous amount of samples. Usually hundreds or even thousands of samples have to be gathered during a process validation program. So that coordination between different groups, both internally and externally, is really challenging and requires a lot of coordination. Really, the project management of this is quite critical. So just probably not so many people are aware, but the number of samples being taken here is quite tremendous. Okay, the last step is the process monitoring. This is after your process is validated, you're doing routine commercial manufacturing, but you still need to show that your process is meeting specifications. You can't take your eyes off the process. You're always looking at the data, even during routine manufacturing. And typically what happens is you want to monitor your process against those critical process parameters that you discovered during the process validation. You want to trend all those. So for example, you would trend growth or product quality and make sure that those are within a certain limit. If you see a certain trend, even if it's within specification, you really want to have a trend that's defined, a definition of a trend that allows you to take a closer look at that. For example, if 5 runs in a row you see an increase in a certain parameter for 5 runs in a row, for example, you may want to take a look at that just to make sure that there's nothing to that trend other than the process variation. And some of the key factors overall of this talk was identifying some key and critical parameters as early as possible in the design phase for the phase 1 projects. Try to identify even transient impacts of parameters. You know, if the temperature goes out even for a few minutes, what does that mean? Or if the pH goes out of range even for 10 minutes, what does that mean? That's going to be very critical going on. The numerous tasks of process qualification require incredible coordination between the client, the CDMO, and all of our external suppliers and testing facilities. It's a very challenging in terms of the data management of that process qualification. And then when you have continuous process verification, the most important thing to realize is that this is your chance to really reduce the number of tests except for the ones that are showing key critical parameters. And you want to show that in your batch records to have a reduced amount of data points. And ideally, you would have this simplified batch record available and show during a pre-approval inspection where they typically like to see manufacturing being done. So all this together, from beginning to end, I think you could show a story to say that your process is robust and reproducible. So thank you very much. Please don't hesitate to ask any questions or contact Avid Bioservices if you have any other questions about process validation. Thank you.